Welcome everybody. I think we can start. So my name is Jean-Michel Marin. I am the Dean of the Faculty of Sciences, and it's a very great pleasure to welcome Professor Peter Reich at the Faculty of Sciences and at the University of Montpellier. Peter is one of the most respected and influential scientists in ecology and evolution. His research has clearly transformed our understanding on how ecosystems respond to climate change. Peter gave us yesterday a remarkable and deeply inspiring lecture. Those of where are present will agree. It was not only scientifically rich, but also profoundly human. Your ability, Peter, to connect scientific insight, ethical reflection, and global societal challenges is quite rare. And we are very fortunate to benefit from it yesterday, and we continue today. So Adam will introduce Peter in more details. I would like to say also that beyond its scientific excellence, Peter is an extraordinary guest. I had the privilege of having both lunch and dinner with him yesterday, and we have a very inspiring and exciting exchange during this lunch and dinner. And you are also, Peter, and this matters greatly today, an ardent defender of sciences at a time where sciences is increasingly under attack in the US. So we discussed about that yesterday a bit, so I have to say that. So Peter is very ardent to that. So today, Peter, we address, compared to yesterday, another fundamental topic. The title is just here. This theme resonates strongly with many of us here in Montpellier, where ecology, evolution, mathematics, physics, medicine interact in rich and long-standing ways. So understanding how simple principles scale up into complex patterns and how complex systems respond to global change, it at the heart of much of our work here in Montpellier. So we are very excited to hear from you. So thank you very much once again, Peter, for being with us. We are honored to once again welcome you here, and we look forward to your lecture. And I give the end to Adam to say more words about you. So thanks again, Peter.
Thank you, Jean-Michel. So thank you, Peter, very much to be with us today, this afternoon, for this second conference. So, donc, mesdames et messieurs, chers collègues, chères étudiantes et chers étudiants, donc c'est encore une fois avec un immense plaisir donc d'accueillir Peter ici, donc comme a pu le souligner notre doyen, donc l'un des écologues les plus influents de notre époque. Donc, avec une conférence consacrée à un thème particulièrement pertinent dans le contexte actuel, donc the strength of the simplicity and the challenge of the complexity, trade-off and scaling in the area of multiple goal and challenge. Donc, on est là ici sur un thème où on met l'accent sur l'accélération des perturbations, sur le changement climatique, la fragmentation des habitats, les incendies, stress hydrique. Donc, et nous devons caractériser finalement ces processus écologiques complexes, voire difficiles à anticiper, à caractériser. Et du coup, et pourtant, Peter, grâce à ses travaux, montrera comment finalement des principes simples, issus de l'étude des traits fonctionnels, notamment, donc des plantes, permettent d'identifier, de comprendre des compromis écophysiologiques fondamentaux et de mieux saisir la manière dont les organismes réagissent, s'adaptent ou déclinent face aux pressions globales. Donc son intervention explorera aussi la notion d'échelle, Donc, c'est-à-dire que de manière dont ces processus observés, par exemple à l'échelle de la feuille de l'individu, peuvent être extrapolés pour comprendre le fonctionnement des peuplements, donc des forêts et même au niveau de la biosphère. Donc, ce qui caractérise encore une fois Peter, c'est sa capacité à, à rendre finalement des notions d'écologie et de les transmettre de façon assez didactique. Donc on a très apprécié son intervention hier dans ce sens, dans ce qui rend encore une fois Peter très original et suffisamment, je dirais, en avant-garde donc en termes de transmission du savoir. Donc nous sommes encore une fois extrêmement heureux de pouvoir bénéficier aujourd'hui de son expertise, de son éclairage, de sa vision des choses, toujours positive malgré la situation, malgré le fait qu'on est dans une dynamique où la biodiversité, les perturbations, les bouleversements écologiques sont de plus en plus, je dirais, parlants, et donc on est vraiment dans une forme d'urgence écologique. Et malgré tout, Peter nous donne toujours ce sentiment qu'il faut toujours agir, qu'on est encore en capacité d'agir et que rien n'est perdu. Donc, Peter, thank you very much for being with us today. And we hope that this conference will be also useful for students, because mostly we decide with the dean to invite you to come in Montpellier mostly for students in order to learn and to have this interaction with you. So once again, thank you and enjoy.
Today's talk will be much more within the realm of a traditional ecological talk. Hopefully that won't be disappointing to folks who might have liked what they heard yesterday. So I'm going to talk to us about whether or not traits and the idea of trade-offs and scaling can help us make sense of the complex world we face, because it's a challenge to do so in a scientific way. We all know how beautiful and diverse our ecosystems across the world are, but that also makes it really hard to understand what's going on in any one of them or to try to generalize across them. And perhaps the attributes of organisms in these systems, whether it's leaves, stems, roots, seeds, or other attributes, maybe they can help us as a common currency to talk about these different systems without having to identify taxonomically every member of these communities and the way they are related phylogenetically or evolutionarily. So the idea behind this is that perhaps plant traits allows us to crosswalk and crosstalk between the way in which environment and evolution influence physiology and fitness, which lead to community assembly, which leads to biodiversity, which leads to ecosystem function, and that plant traits are related to the processes at each of those different kinds of processes and scales, that maybe by using plant traits we can accelerate the way in which we can think about those and generalize about those across different systems. At least that's the premise and the idea. And we'll see whether you buy into it or not. So this whole idea of trait-based ecology really asks about how structure and function of communities and ecosystems emerge from functional properties of organisms, or at least that's one aspect of trait-based ecology. And at the bottom, you can see just here in Montpellier, easily from the last couple of days, many names of people who work on this very theme here, and I'm sure I left off many, many other names. But I really think of trait-based ecology and its value as a substitute or alternative to species-based approach. Rather than having to understand everything about a maple or an oak, we can instead think about what's their chemistry, what's their shape, what's their size, what does that tell us about their ecology, physiology, and interactions with other organisms. So one way to think about this is, if you think about a species like Picea glauca, a white spruce in Canada, and a kingbilly pine, an ancient conifer in Tasmania, that both have leaves that live for many years, that have low nutrient concentrations, do the fact that they share those attributes tell us anything about their ecology, the way they interact with other species, the role they play in these systems? And that's the test we want to make with trait-based ecology. So I'm going to share today some ideas about whether or not there are fundamental rules in trait-based ecology, and try to at least address, and likely not answer, but at least address these 5 questions. First, are there a limited range of successful designs? In essence, are there really plant trait strategies, or can species have every combination of trait under the sun? Second, are there predictable rules for the way traits vary along environmental gradients, whether they're temperature, moisture, or other? The next, are there predictable rules for the way traits act as drivers of community assembly? And then fourth, do traits allow us to link leaf or root or stem to individual, to community, to ecosystem processes? And last, do these traits tell us anything about ecosystem function and the response of these systems to global environmental change? In the next 45 minutes or so, I'll touch upon at least these 4 concepts in theory at different times during the talk. A lot of this is based on trait trade-offs, the idea that you can't be good at everything, and the traits that are good under certain conditions aren't useful under others, and that you can array most species along some kind of fast versus slow lifestyle. And that's what I'll start with in the talk, but also we'll touch upon these 3 other concepts during the talk. The whole idea of ecological stoichiometry, that the ratios of elements in our tissues and in our bodies influence how we behave ecologically. Then when we switch to thinking about systems rather than individual organisms or species, do the average traits of a community tell us something about their function as an ecosystem? Or instead of their average traits, is it the diversity of those traits that drive biodiversity ecosystem function? So I'm going to start with leaves as a test bed for theory, the idea that maybe there's a trade-off between productivity and persistence for leaves, just as there might be for entire individual organisms. And some of the ideas embedded here are this notion of fast versus slow strategy, which in some ways is an update of R versus K strategies. We could also think about multi-dimensional trade-off envelopes. It's much easier to show a bidirectional plot in a 2-dimensional flat screen than an n-dimensional one with n being 6 or 7 or 14. But when we think about traits, we're really thinking about multi-dimensions. And lastly, if these trade-offs exist, do they exist within and among communities? So in this cartoon hypothesis here, we could say that maybe this notion of productivity versus persistence creates a viable fitness envelope where all species, in order to be successful, have to live somewhere in this gray shade, in this gray shaded area along this line here, going from the so-called tortoises to the hares, going from ones that are good at persistence to ones that are good at being productive, with the idea that maybe outside of that trade-off envelope, you can't exist down here. You could engineer a plant that would do this, but if you're not persistent and you're not productive, you're not going to be very successful. On the other hand, maybe it's biophysically impossible to construct a leaf or an entire plant that's both productive and persistent. And so that's the idea we're testing here. So my first, or one of the first tests I made with this was combine data for 100 species I had and colleagues had measured in the field at 6 sites from the tropic to the tundra, with 100 data points for species around the world from the literature. And we looked at whether or not there were links between chemistry, structure, and function. And what we found using this 3-dimensional plot was that all the species were arrayed along a plane that linked these 3 traits, so that the productivity in terms of photosynthetic capacity of species was higher when they had thin leaves with high specific leaf area and high nitrogen concentrations, with those hares of the fast species up here and the tortoises down here. And that this was true for species no matter which biome you were from. And that you might say, well, why don't all species have high fast traits? Because you have high capacity, spelled wrong here, photosynthetic capacity and growth capacity, but there's trade-offs because that comes with high costs. You use a lot of water, you lose a lot of energy in dark respiration, your nutrient acquisition to drive that photosynthetic capacity is expensive, and if your leaf turns over rapidly, you have high replacement costs. And so when conditions are good, maybe this high photosynthetic capacity is a great trait to have, but it may not be fitness generating under all conditions. So that was our first work, our own, and then combining work with people like Ian Wright and Mark Westerby from Macquarie University at the time, and people from around the world, including Eric Garnier, who I think is somewhere in this room, and probably some other people in this room. We put together data from 175 sites, expanding from our original 6 sites that we had done, with 2,500 species, and asked basically the same question about whether or not there were these trade-offs that allowed us to identify what we called then a leaf economic spectrum, and published a bunch of papers about this. And basically, the answer we came up with is exactly the same in the earlier tests. Species within and among systems fell more or less along these trade-off envelopes at the leaf level. And here's one way of showing that. I've grouped the species into 5 different kinds of biome life forms, and looking at the bivariate relationships between leaf nitrogen and leaf lifespan, photosynthetic capacity and leaf lifespan, and photosynthesis and leaf nitrogen, we see common relationships. The slopes are not exactly the same, the intercepts are not exactly the same, but in general we see similar trade-offs in all of these systems. So really seeming to find something that holds under all different contexts and in different kinds of systems. So fast forward yet another 7 to 9 years, and teaming up with more like 50 or 100 scientists from around the world, led by Jens Katke, and Sandra Diaz was a key player in this as well as probably a number of folks in this room. We built the TRI database, which has grown and grown, has far more than 7 million records and 150,000 taxa now, and is the go-to resource for people trying to get plant traits in most contexts. And so one of the papers that came out of this was led by Sandra, where we used 6 traits for 2,200 species, going beyond just leaf traits and including seed size and stem density and plant height. And you can see representing in the dark green much of the biogeography of the planet and much of the so-called Whittaker space on the left. And we found many things with these data, but to me the most interesting one was that actually 98% of possible trait combinations are not successful. They don't exist in nature. So this is a 6-dimensional trait hypervolume, which makes my mind hurt to try to think about. But the way I like to think about this is that of the 100% of 6-dimensional trait space, only 2% of that is filled. So, and that's because there are correlations between functional traits that limit the number of possibilities. So this idea of traits and trait trade-offs really does create or does define the multi-trait space in which plants are successful. So we can ignore 98% of possible trait combinations because they simply don't exist in nature. And so traits do help us define and constrain the way in which plants and their interactions with other traits, plants and their traits and their interactions with other traits work. So we've seen that there are a limited range of successful designs, and that this holds up in every single ecosystem around the world. There are kinds of trait combinations that simply aren't successful or aren't possible. So the next question, we'll move on to number 2, does that vary with the environment? How do these traits vary with the environment? And I'm going to give, I think, 3 examples here at local and regional scales. The first is at the Cedar Creek site, where some of our other experiments are. We also have a long-term fire frequency gradient, which actually we think is the second largest and second longest running fire experiment in the world. Since the 1960s, we've been running fires, either no fires, 1 in 10 years, 1 in 3 years, 1 in 2 years, 3 in 4 years, across several hundred hectares. These are prescribed fires in the spring. And this low to high fire frequency gradient has led to a spectrum of vegetation from closed canopy forests on the left to shrubby woodlands in the middle to open savannas on the right. And so we can ask, what do the plant traits and the community-weighted mean traits look like across these gradients? What we find is that there's a sharp gradient where under low fire and high soil nitrogen, which is dominated by forests, we tend to have high fast plants on average, with the shift going to slow plants with low nutrient concentrations and thicker, denser leaves at high fire and low soil nitrogen, which we see at the high fire end of the gradient. Now, what's really interesting here is that we also see how differences in species composition and plasticity within species contribute to the overall gradient in traits. The palest orange color there are the functional group means, and that slope is the shallowest, meaning that as you go from the forest end of the gradient to the high fire grassland end, traits like nitrogen go from high to low, but that as you shift to the species means or the individual means, the slope becomes steeper because plasticity is within species is pushing the gradient up here at the forest end of the gradient and pushing you down here at the other end. So this gradient in this particular case of community weighted mean traits is driven by compositional differences across the gradient, but also plasticity within species. And this is work led by Adam Pellegrini, who's just moved to Stanford recently. So that's one gradient, a local gradient in fire and associated nutrient availability. How about a larger scale gradient and a different kind of trait? Here we're asking whether tree plumbing or the hydraulic architecture differs along rainfall gradients. This is work led by Sebastian Fauch at the Hawkesbury Institute in Australia, where he sampled tree hydraulics along two large biogeographic gradients in eastern and western Australia and measured hydraulic traits and wood density. So this is a test among eucalypt species, around 30, and as well there was some within species variation as well that followed the same general pattern. And what we found was that there's a very strong variation in hydraulic architecture within this genus along the dry to wet continuum, where in the wet end of the gradient, you can see species like Eucalyptus regnans have vessel diameters that are much larger, but are more prone to cavitation. And as you go towards the dry end, the vessel diameters decrease, and slower growing species have narrower vessels, which protect them from cavitation under drought. Of course, there's always trade-offs, though. Those species, because they have those narrower vessel diameters, don't move water as quickly or in the same velocity and capacity, and so their total photosynthesis is restricted by that lack of ability to move water quickly. So you can either be hydraulically safe or you can have the capacity to move water and carbon quickly, but apparently you can't do both. And within this genus, you see quite a remarkable and clean variation in these particular traits along this climate gradient. So I'm going to shift from thinking about traits along the environment within or among species to thinking about traits as drivers of community assembly. And here I'm going to give 3 examples. The first was a study led by Joe Wright at the famous BCI 50-hectare mapped plot in Panama, where he and others had been not only keeping track of the growth and survival of, I think, more than 360,000 individuals for more than 20 years, of more than 100 species, but also measuring the average traits of more than 100 of these species to see whether or not traits could explain the long-term dynamics of this community. And Joe invited Ian Wright, who's no relation to Joe, other than they're both great scientists, and me and a number of other people to help him with this paper. So we went down to Panama for a couple of weeks and threw the data around and came up with this story. So what we see, first of all, is that there is a trade-off between maximum growth rate and survival of individual species at the individual tree level. So those that grow fastest have the lowest survival, and those that grow more slowly tend to have much higher survival over a 5-year period. So we see that this growth versus mortality trade-off is strong at the level of individuals for these set of species. Well, what about traits like plant traits? Do they explain what's happening along this gradient? And the answer is yes, up to a point. So we only measured, and we had this discussion earlier today, that we don't always necessarily measure the traits we really want. They had 4 traits here and 2 of them explain 40% of all the variance in growth mortality across this gradient. Those were wood density and leaf mass per area. And in general, the high tissue density, the dense wood and denser leaves were associated with the species with high survival rates, but low growth rates, which is consistent with other studies. Now, when we first started looking at the data, Joe Wright was very disappointed because he pointed to Ian and me and said, you guys have been telling me how great traits are. We only explained 40% of the variation, across all these species across 23 years. That's not very good. Whereas Ian and I, we were like flabbergasted, like just 2 traits at average value for species tells you 40% of everything you need to know about the system. And so, I think of it, the glass is really half full. Traits don't explain everything, but in this case, they tell us a lot about the structure and function and interactions among these species and the way they are able to co-occur in this system because of this heterogeneity in light and water within the system. And so I think this, to me, is a lesson that we should think that traits are helping us get down the road towards understanding these systems, but we shouldn't expect them to explain everything. Whoops, put that up too late. But so the next example is going back to the Cedar Creek, and in addition to the fire experiments and the global change experiments, we have some pretty large-scale, long-term nitrogen addition gradients just in grasslands. And so here I was asking, using data from a 20-year period, whether or not species with fast traits, meaning high photosynthetic rates in this case, versus low photosynthetic rates, were differentially successful in high nitrogen versus low nitrogen plots over this long time period. What we see was a very consistent trade-off, that the relative abundance in the second highest N availability was strongly inversely related to the relative performance at the low N availability. You can see that on the left there. And that the species that do poorly under the highest nitrogen tend to have the lowest photosynthetic rates, and those that have the highest photosynthetic rates do well in the higher nitrogen. The reason I put the second highest nitrogen availability is that we're looking at the photosynthetic rates in the highest, so we're trying to have the data be independent. But in general, we see species can have fast or slow traits, and the fast traits help in resource-rich conditions, and the slow traits make you more successful in the low nutrient conditions in this context. The last example is going back to the tropics to Australia, and this is work I was not part of, but I was very impressed with it when I went to visit there last year. The John Dwyer lab in Queensland has been looking at an amazing number of species, 285 rainforest species along a 600 to 2,500 millimeter rainfall gradient in eastern Australia, and measuring a number of traits, including hydraulic traits. And what they found was that these hydraulic traits really explain a lot about where you find these species most commonly. So if you look on the left part of the figure on the right, what we're seeing is the species habitat affinities, whether they're most common in the wet up on the top, I think this is dying, or they're more common in the dry, which is on the bottom lower left, and that the hydraulic strategy matched what Sebastian Fauch had found within eucalypts, in that those with small vessels and high wood density were most abundant in the dry end of this gradient, and the 285 species, those that were found in the wettest gradient tended to have large vessels and low wood density. So we see this association of traits with the occurrence of these species across this large gradient in this complex rainforest system. So I'm going to leave off thinking about community assembly and instead now think about whether or not traits matter to the way communities and ecosystems function, thinking about the community as a whole rather than the way the parts assemble themselves. And here I'm going to talk about 4 things, or at least touch upon 4 things, but I'm going to jump right to the trait functional diversity idea involved in the way in which biodiversity might enhance ecosystem function. So I showed this figure yesterday. This is from a very large compilation of data from more than 80 countries around the world, in which we were able to look at productivity in relation to variation in tree species richness within regions, and everywhere on Earth, as your tree species richness is lower than the maximum for your region, productivity was lower. And as I mentioned yesterday, there are dozens of tree diversity experiments that have very similar shaped curves for biodiversity productivity relationships. And we also have seen in grasslands, in both observation experiments, very much the same thing. This is the BioCon experiment that we've been running for 27 years in Cedar Creek, in which we grow plants in different numbers of species richness levels from 1 to 4 to 9 to 16 at both ambient elevated CO2 and ambient enriched nitrogen. And there's 371 plots, 2 by 2 meters. As I mentioned, the ambient elevated CO2, ambient plus 180 parts per million, soil nitrogen ambient plus 4 grams, and with 4 levels of species diversity drawn from 4 different functional groups. And Sarah Hobby is shown there, one of my long-term collaborators on this project. And what we found here is that there were strong effects of biodiversity, of CO2 and of nitrogen in interaction, such that diversity increased productivity, and increasingly over time under all CO2 and N levels, and CO2 also had big impacts on productivity in these systems. So here's examples of grasslands and forests. There's lots of outcomes of biodiversity, but none of these experiments really so far that I've shown you have much evidence to really explain the mechanisms. And I think that's true. There's probably 100 biodiversity experiments and almost none that really allow you, we wave our arms and try to explain what we found, that it's consistent with theory, but very few have actual evidence for any of these kinds of mechanisms of biodiversity effects. And this is by no means a complete list of the possible mechanisms. Many of them involve what I think of as niche differentiation, species differing in their resource uptake potentials, their resource use, their phenology, where they exist in space or time, or the thermal optima. They also can facilitate each other by protecting them from too much wind or too much light. There's also longer term things such as resource feedbacks with increased soil fertility. Then I put way down here the selection effect, which is whether a particularly productive species shows up in a system, and it's way down in this list because almost every long-term experiment has shown that selection is not the main driver of biodiversity effects, it's complementarity. And there's a couple of other ideas down there about what can lead to biodiversity positively influencing ecosystem function. And so what's the role of traits in this? Well, the example I'm going to give you here, and I'm going to spend a bit of time on it because it's a bit complicated, is one of the ident experiments, a family of high-density tree diversity experiments. This one had 6 species from North America and 6 species from Europe planted together at Cloquet, Minnesota. Very close spacing, 0.4 meters, 192 different plots. Most of them either monocultures or 2 species mixtures. 9,000 odd species, individuals planted at the start of the experiment in these multiple blocks. And what we looked at here was, rather than just varying species richness, we had many, many of these 2 and 4 species plots in which we had a stratified sampling in which we intentionally put together species that were functionally more similar or functionally more distant to try to see whether even holding species richness constant did functional diversity matter. Just some pictures of the 1st through 6th growing season. We're now in the 15th year of the study or so. So Laura Williams, who was a PhD student and then a postdoc of ours at the time, and is now at the Hawkesbury Institute in Australia, one of the things she wanted to ask was whether spatial complementarity contributes to over-yielding. And so she wanted to look at whether the trait values of individuals in neighborhoods, those trait values of their crown shape were varied by who they were next to, and whether that led to over-yielding at the system scale. At the start, I discouraged her from doing this because I thought it wouldn't work. I thought it'd be too difficult, but she was very hardworking and persistent. So what she did was she measured for 328 trees, and this is actually at a different ident experiment where she did this work. She basically characterized the crowns in this way, kind of low tech, but hopefully meaningful, to look at where the crown was, how widely it was spaced at any height, and where that crown was on the height of the tree. And And so the species differ in average crown shape. It doesn't matter which one you're looking at, but you can see they differ in height and in crown shape. But what was really interesting is whether or not, when you look at the predictions from monocultures, this is the crown shape of Betula peporifera and Quercus rubra shown individually, whether they're together, do they have overlap that's different than they would have predicted from their monocultures. And what she found was that if you go from left to right, these are mixtures ordered by increasing functional dissimilarity in growth rate. So on the left, you have species that have the most similar growth rate in monocultures to the ones that differ the most. And you can see that on average, crown complementarity, or the degree to which they don't overlap, is generally higher on the right than the left. And that this actually is related to functional dispersion, or how different they are in growth rate, shown in the left panel here. And more importantly and interesting is that this crown complementarity, the CCI observed in 2 and 4 species mixtures and 12 species mixtured, was strongly related to whether the biomass overyielded. And what we mean by overyielding is whether a 2 species mixture on average was their tree growth more than you'd predict from the 2 monocultures, or if there's 4, was greater than you'd predict from the 4 monocultures. And we see that there's a strong correspondence between how much crown complementarity or lack of overlap there was and the biomass overyielding. So this is one of the few cases where we actually have evidence for the way in which, in this case, shifts in plasticity lead to overyielding of species in diverse communities. Now, she was interested in taking this another step farther, asking whether or not overyielding would be due to greater light interception or efficiency of light use. And so she actually measured light interception, and we inferred efficiency of light use. And what she found is that overyielding in biomass growth in these mixtures was related both to overyielding in LAI, meaning having greater leaf area index than you'd expect from monocultures, but also in intercepting light more than you'd expect in monocultures. And so the NE is the net biodiversity effect on stem biomass growth. You can think of it as overyielding. Anything above 0 is overyielding. And the NE for FA-PAR is the overyielding of light interception. Anything above 0 is overyielding in light interception. Same for LAI. And you can see there in all of the 4th, 5th, and 6th year of the study, there were positive correlations between the overyielding in intercepting light and in building canopies, and in how much growth these trees were able to put on. So again, ways of looking at crown shapes, light interception, and the roles those play in the biodiversity effects on overyielding in this particular study. So we see there's lots of evidence that biodiversity leads to higher productivity in systems, and there's increasing theory around it, but we're still in the early days of trying to test directly the way in which those mechanisms work. I'm going to move on now to the last part of the study, of the talk, in which we talk about whether or not there are predictable rules from traits for responses to global change. And I'm going to give 4 examples here. The first is intraspecific within species. The second is cross-species, but again at the individual plant level. And then the 3rd and 4th are at the ecosystem level, looking at the response of the whole system with empirical data, and the last one using some trait-based modeling. So intraspecifically, I'm going to talk about work with Pinus sylvestris that was led by my colleague Jacek Oleksin from Poland, in which we actually measured needle traits of Scots pine along a large climate gradient in Sweden. And you can see the needle sampling points, about 70 of them across Sweden. You might ask, why Sweden? Well, the reason is that Jacek fished out from a garbage bin at the forestry library at the University of Minnesota this book from 1917, which was a Swedish State Forest Research Institute report. Jacek was always dumpster diving for old data and old literature. And this actually had in this report in German and Swedish, which we were able to get translated by colleagues and Google Translator, it actually had data on needle lengths and needle longevity for Scots pine along this exact same gradient. So we were able to go back by coincidence 100 years later and see whether or not the needle traits had changed everywhere or anywhere in 100 years as climate had warmed and precipitation had stayed roughly the same. So Jacek organized a number of campaigns of traveling across Sweden. We don't have pictures from 1916. If they exist, they're lost. And this is very low-tech sampling. Use pole pruners, take branches from high in the canopy, sort them to needle-age classes, scan them, do all those kinds of things across this gradient in Sweden. It was great except for the bugs. And what we found is that everywhere in Sweden, all the pine forests have faster traits than they did 100 years ago. And I can walk you through this diagram. In each time period, whether 1915 or the 2015, you can see that needle length is longer in southern Sweden and shorter in northern Sweden, which is consistent with trees that grow fast in southern Sweden, more slowly in northern Sweden. Similarly, the needle longevity went from about 2.5 to 4.5, from about 3.5 to 8 in 1915, because slow-growing trees in northern Sweden keep their needles longer. And those biogeographic patterns across climate gradients were very similar in 2015 as in 1915, but the entire lines shifted because it had become warmer in 100 years. So basically, you can think of the lines, and actually we have in the paper, if you put temperature down instead of latitude, they don't exactly cover each other up, but they come together. So we're really just moving in climate space from 1915 to 2015. Now, actually, been pretty disappointed that not very many people have read this paper or cited this paper, and we actually thought it was very, very exciting to actually find a very consistent fingerprint of climate change across the forests of Sweden. And I would bet $1 million if I had it to bet that we would find exactly the same thing in Canada or Siberia, because the fundamental question is the same. And so within a species, we see strong evidence that the traits have shifted considerably in 100 years. And so what we then do with these traits and how they tell us about the behavior of the system is something else. But I've taken out of the talk, so I wouldn't go too long. Another paper we wrote looking at variation in needle longevity and other traits in Canada, in which most of the global models assume that needle longevity is the same everywhere in the boreal forest. And actually the models use a turnover rate of needles of about 3 years, which is too low everywhere for spruce and is also too low in many places for pine. But that by incorporating the reality of needle variation within species across these large climate gradients, the models behaved much better and predicted eddy flux at a number of sites much better. And so understanding trait variation can help us understand the way in which these systems both are responding to long-term climate change, but the way in which they might be functionally behaving differently now than in the past. The next example is a cross species, but in a given experiment. This is the before warmed experiment in Minnesota, where we're warming by 1.5 or 3 degrees Celsius, both above and below ground, and removing rainfall in half the plots by removing 40% of the rainfall in the growing season in half the plots. And the 2 slides I want to give here show that the effect size on growth and survival, shown on the y-axis, anything above 0 means you're growing or surviving better, below 0 means you're doing more poorly, was related to the way in which these global change treatments influence photosynthetic rate, measured rate in the field. Again, anything above 0 means they're photosynthesizing more under the warmer and or low rainfall conditions. Anything below 0 means you're doing more poorly. And you can see that the species treatment combinations where photosynthesis was increased are the ones where survival was better and or growth was enhanced. And those where photosynthesis was poorer are the ones where survival was poor or growth was poor. So we see a link between leaf level physiological response and growth and survival response. And we can go one step further and look at the way in which infrequently measured trait can tell us about these photosynthetic responses. And here we see that the change in photosynthesis due to this warming and rainfall change is related to the sensitivity of stomata to soil moisture. So basically, under ambient conditions, species that tend to close their stomates earlier when the soil is less dry, are the ones that have negative responses photosynthetically and growth and survival to warming. You can translate that into stomatal limitation of photosynthesis instead, which is on the right. But in general, here's a trait that you can't find in tri, which is sensitivity of stomatal conductance or stomatal limitation of photosynthesis to soil moisture, but explains a lot about the way in which species differ in their response to global climate change. The last two examples are at the system scale. The first involves the response of terrestrial ecosystems to CO2 under the diversity of nutrient availability that exists in nature. So the question is, does nitrogen or phosphorus limitation cause decreasing productivity under rising CO2? And this is important because Earth system models predict large carbon sinks even in the tropics. And if that's not true because of low phosphorus and nitrogen availability, that's a problem. There's been a bunch of studies, including our Biocon study, looking at CO2 effects under ambient versus elevated nitrogen. And what we found over many years now is that CO2 fertilization of productivity in our grassland exists even at low nitrogen, but we see twice the response under higher nitrogen. So across a continuum of nitrogen availability in nature, we're going to see in the real world variation in the CO2 fertilization effect. And these are 4 of the many papers in which we've looked at this. But I want to spend more time talking about phosphorus because we have far fewer experiments with phosphorus limitation. And here's the first of now 2 large-scale, mature tree scale face experiments. This is the eucalypt face experiment in Australia, where for more than 10 years now, plants have been 30-meter tall trees have been exposed to either ambient or elevated CO2. And the question here was whether these low phosphorus systems would constrain how much elevated CO2 increased the growth and productivity and carbon in these systems. And the answer was, well, it not only constrained it, it completely eliminated it in this eucalypt free air CO2 enrichment experiment. And it shown Dave Ellsworth and Belinda Medlin, 2 of the leaders of this study. We see absolutely no positive response to elevated CO2. It's a system that's got very low phosphorus in the soil and the leaves, and it's also somewhat nitrogen limited. And so what we're seeing is that there is a big impact on the CO2 fertilization. Here's data from early in the experiment, where in the first 3 years, the darker of the paired bars is under elevated CO2, the left bar is ambient. There's no evidence that above ground NPP or any element or component of it is greater under elevated CO2. The bars look the same for all the subsequent years. And then we've also done some full ecosystem carbon and phosphorus cycle assessments led by Mingkai Jiang. And both of them showed that the fate of carbon in the system is due to the fact that although the photosynthesis is increased by CO2, it goes into the soil and comes right back out of soil CO2 flux. And so the system is cycling carbon faster, but it's not holding on to it, and it's not even using it for any growth. And the reason for this, and it's the second of the 2 papers by Mingkai Jiang that he led, is that the microbes outcompete the plants for any phosphorus, and it's really, there's so little phosphorus they're not able to use the extra CO2 to grow. They can photosynthesize and take it up, but they don't have phosphorus to build more tissues, and so it basically just gets sent back to the atmosphere. Now, this is the dark green shows the world's rainforests, most of which are phosphorus limited. Does the Uke face experiment mean that they all have low carbon sink strength? We have no idea. You need 10 experiments like this because my hunch is that you'd see low carbon sink strength, but you would not see 0 carbon sink strength from elevated CO2 in most of these systems because they're not all as phosphorus limited as the Uke face. But without actually having those experiments, all you can do is conjecture or build models to try to test that. And that'll lead me to my very last example, which is in the Terracon experiment, which I mentioned yesterday. It's a sub-experiment in Biocon, 48 plots that were planted with 9 species from a 16-species pool to all 16 combinations of elevated, ambient elevated temperature, ambient reduced rain, ambient elevated CO2 and ambient and enriched nitrogen. And what we're looking for here is whether or not these drivers individually and or together influence productivity and many other aspects of carbon and nutrient cycling, as well as biodiversity and soil communities as well. And the thing I want to share here is that we, working with Lei Zhang at Berkeley National Lab and Bill Riley there, we parameterized a trait-based model called EcoSym with C4 grasses, C3 grasses, C3 forbs, and C3 N-fixer traits. And I was actually very surprised that the model predicts well the responses over 12 years to these multiple global changes. There's a lot of different contrasts shown there. The top 4 under A, well, the left column is effect on NPP. So anything above 0 means NPP is increased by treatments. Below it means it's decreased. On the right is the effect on soil CO2 flux. Same, above 0 means it's increased. A and B show the individual drivers, temperature, rain, CO2, or nitrogen alone. And the blue is the experimental results, and the orange is the model. And although the model is by no means exactly matching the results, it gets the direction right, and it's pretty close to the observations in most cases. And it also gets most of the 2- and 3- and 4-way interactions shown here approximately right. So here we have a model in which, by parameterizing with the traits of the species from this experiment, under ambient conditions we took those traits from, it was able to predict the way in which they interact and either grow more or grow less or grow the same under different combinations of global change drivers. So it gives me some optimism that, models, the famous saying, all models are wrong, some are useful. Maybe this is one of those that is useful, and we can continue to try to understand in the model framework why we're seeing the responses we are, because the model includes more mechanisms than we can actually measure. I think with that, I've gone through this quick short course in trait-based ecology that actually matches. Eric Garnier showed me the book that he and others are putting out this year and that I might participate in, which is in great detail going to go over all of the things I talked about today. So I gave little snippets or snapshots of some of those from my own work and things I'm aware of about everything from the range of plant strategies and whether or not there's a limited range of successful designs, to how traits vary across environment, to whether traits can help us understand community assembly, whether they can help us understand the way in which individuals, communities, ecosystem processes are linked, and whether or not they can tell us anything about the way our ecosystems are going to respond and our species in the changing world that we have today and in the future. So if we have time for questions, I'd be happy to entertain them.
Thank you very much, Peter. Very inspiring and interesting talk. I have a question regarding the ecosystem functioning part and the role of biodiversity and functional threat as driver. So you present us nice case where the functioning of ecosystem, mostly productivity, increase with the number of species present, and you dig into the threat of the species to explain the pattern. But I am wondering if you already investigate also the negative case. I mean, there is a huge variation in this effect of species richness on productivity, and sometimes the more species you add, the less productive the ecosystem is. And or even in a biodiversity experiment, I mean, you can have mixtures that produce less than monoculture, for example. And do you have any idea of the mechanism or the process that can be at play in this negative case?
Great question. In our experiments, when we put together the plots that have the greatest number of species, we actually always see much greater productivity than the average monoculture, not necessarily the most productive monoculture, but we do see some low diversity combinations, 2 species or 4 species, that do underperform their mixtures. And we actually haven't investigated very much what's going on with them, whether there's some kind of, obviously, negative interactions between the species. But I think you're pointing out that describing and characterizing the average nature of biodiversity ecosystem function relationships was a great start, but now we need to take that apart to understand, when do we going to see that? When can we enhance it? When will that not work? And so I think you're pointing us in a good direction there. And actually, what we actually see in our experiments, including not just the ones that we're running, but the ones that are in the tree diversity and grassland diversity experimental networks, is that most of the variation is in the monocultures. The mixtures generally perform well and have low variance, and the monocultures vary tremendously among species, but also over time. And so maybe we need to start thinking about the links between stability and productivity in those kind of contexts. I mean, and I could talk to you for hours about potential reasons why certain mixtures don't work well, but that's just conjecture on my part.
Thanks a lot for this really great talk. Bit following up on that, what's your guest's idea on how this fertilization effect will be counterbalanced by kind of a loss of stability of the ecosystem with climate change? So we might produce more, but we can fall from higher places.
Yeah, so you're basically asking whether or not climate change is going to, by reducing diversity, is going to really impact the gains we get from diversity?
Yeah, and also if it, yeah.
Yes, no, I think it's a huge thing because and actually I was part of a paper led by Akira Mora about 4 years ago in Nature Climate Change, which did some global modeling of this. And there's strong synergies between diversity and stopping climate change because more diverse systems will store more carbon. But the more in which we allow climate to change and that erodes our diversity, that's going to erode the ability of systems to store carbon. It's going to be a negative feedback, well, positive feedback, but in negative consequences for climate. So I think it's a big concern.
Yeah, thank you very much for your talk. So I was wondering, what do you think is the most useful for predictions? Trait means or trait plasticity?
I don't think we know the answer yet. I think some combination, because plasticity by itself, and it depends on what your question is, but across species it vary a lot. Trait means probably are going to tell you more, unless you're also asking how they behave and under a very large gradient of context, in which case then the plasticity may also come into play. And so I think that so I think it really depends on exactly what context you're looking at.
Yeah, in response to future environmental changes, maybe we have overlooked trait plasticity, why it could be a major driver of response to environmental changes, and maybe it's something we have missed too much in trait-based ecology.
I think it is, but I think depending on which trait, some traits, almost all species actually are equally plastic. Like for instance, changing SLA in relation to light. All species do it. There's some variation in how they do it, but it's not huge. I mean, yes, conifers do it less than angiosperms, but there are other kinds of plasticity in which species vary all over the map and how much they shift. So I think there's a lot we still don't understand. And there's some things that aren't necessarily going to be plastic, like you can't change your vessel architecture very easily. That's not a particularly plastic trait. And so it also depends on which traits are the most plastic. Probably one of the most plastic ones that we have, I think, almost no data on is the relationship between species that are mutualists, especially things like mycorrhiza for the vast majority of species that are mycorrhiza. If there's 300, if you're an ectomycorrhiza and you have 300 potential partners, which one of those partners you shift to, depending on context, may either allow you to perform better or worse, depending on the context. And we have almost no data in any comprehensive way about that kind of variation. And so I think, yes, there's so much about, and to me that fits as plasticity. But
thank you for a great talk and very inspiring. And it's just a question maybe to put things back in perspective. I mean, with your amazing experience asking these questions at various scales, if you could do it again today, like go back, what would you change, or what would you be most excited to ask?
Whoa. That's a very challenging question because it depends on how narrowly I confine myself to, if I confine myself to what would I do differently in trait-based ecology, it might be, it depends on whether I can change time. If I'm starting now, I do something different because with the tools, when I started, our ability to even measure mycorrhiza, for instance, was, you had to do everything by hand. There were no DNA to do those kinds of things. And so I think some of the things that are very exciting are looking at microbe interactions, plant-plant interactions, plant-micro interactions, because I think there's so many degrees of freedom for those kind of interactions to influence and drive systems in all sorts of ways, that there's lots of degrees of freedom for that. And so to me, that would be a very exciting thing to do as far as starting now. But going back to some of the questions I got yesterday in my talk, given the fate or the shape of the world we're in, maybe I would go into policy and not into science. It's more boring. To me, science is exciting, but maybe instead of making one drop of water into helping the planet do better, I could do two if I had gone in that direction. But
I had a question kind of related to the talk yesterday also, but based off of what you were saying about the fact that carbon, even if there was more carbon for the trees, that phosphorus was a limiting factor. And so if we're thinking about these carbon banks, these carbon land banks, could it be a potential solution to helping trees store more carbon by aiding in the giving more phosphorus? Like if we use some of the waste that we have, like we have a lot of phosphorus and elements that come out of our waste. If we add this to all these forests, could that be a potential, I don't know, could that help something, or do we not know enough yet?
well, in a generic way, yes, the answer is yes. That one of the things we talked about yesterday is that one way to try to have a just management for trying to maintain and preserve and enhance tropical forests, in particular in the global south, involves involving local people and encouraging them / allowing them to use the forest to also produce products, and especially if those products are like things that have a long lifetime rather than being turned into pulp and paper or something like that. And so if as part of that, fertilization was part of the management of those systems and can allow them to grow larger, faster, and hold more carbon, if it's done in a way that more carbon stays out of the atmosphere, then it is a win-win-win. Doing that in every particular context is going to depend on that context, whether you're replacing a rubber plantation or a palm oil plantation with some kind of agroforestry. The devil's in the details, because then you're really talking about management on a parcel by parcel basis. But I think, yes, to my mind, the answer is yes. We can hope that nature can help us, but we can also help nature help us. But it's not going to be easy to even theoretically think about exactly how to do that.
Thank you, Peter. Again, let me thank Adam to organize this meeting with Peter, and also the communication office of the Faculty of Science, who was very efficient in organizing these two conferences and also the roundtable. So Peter, thank you again to come in Montpellier.
Thank you all for coming.